complete this chapter, and I will take this too in this week. Denaturation of protein. What does this mean? So first of all, I'm going to write it and then I'll explain. It's very easy. So please write with me. Wait a minute, I'm not concentrated like this. Okay, so let's start. Whenever physical condition like temperature or pressure, physical conditions like temperature, pressure, or chemical condition like pH value of protein changes, then protein loses its identity. Then protein loses its biological activity. Biological activity. This is known as denaturation of protein. This is known as denaturation of protein. For example, I'm going to take example like milk converting into curd. So first of all, the example we can take curding of milk by changing pH value, by changing or by change in pH value. pH value of milk. Second, we can take example of coagulation of egg white. Coagulation of egg white. That means we are talking about albumin. By changing temperature, by changing temperature, that means boiling. When we boil egg, that is the condition. So it's very easy to understand. Basically, what is denaturation of protein? Whenever there will be physical or chemical changes, like physical changes like change in temperature or pressure, or chemical changes like change in pH value, then protein loses its biological activity, and this is known as denaturation of protein. When curd is prepared from milk, you can see it has change in chemical condition like change in pH value. So this is the denaturation of protein. And we can also take example of egg. When you are going to boil it, there will be coagulation of white part of the egg, that is called albumin, and it is basically example of denaturation of protein. Is that clear?
Yes, sir.
Okay, thank you. Please write it and let me know when we're done with this. After this, which chapter we are gonna start? Please let me know.
Sir, phenols, alcohol, phenols, and ethers.
We are done with haloalkanes, right?
Yes, sir.
Okay.
Yes, sir. Finish.
Very good. Sneha, are you done with this?
Yes, sir.
Okay, very good. Now we are going to study about nucleic acid to see what is basically nucleic acid. So first of all, write with me, then I'll explain. Nucleic acids consist of polynucleotides. Polynucleotides. And what is polynucleotides? So I'm going to define it. Polynucleotides. Poly, that means many or several, you can say. Polynucleotides. So basically polynucleotides consist of nucleotides attached with each other. Nucleotides attached with each other by a linkage known as phosphodiester. Known as phosphodiester linkage. So basically, there are several nucleotides that are attached with each other by a linkage that is known as phosphodiester, or we can also define it in the way, in other words, like in polynucleotides, in polynucleotide, Nucleotides are attached. Nucleotides are attached with each other by phosphodiester linkage. Phosphodiester linkage. So several or many nucleotides are attached with each other with the linkage that is known as phosphodiester, is known as nucleotides. And basically nucleic acid consists of this polynucleotides. Please write it. Then we are going to nucleoside, define nucleoside and nucleotide. Let me know when you are done with this.
Yes, sir. Finish.
Are you also done with this?
Yes, sir. Done.
Same. Okay, thank you. So see, it will be more easy if I'm going to write in a very simple word. You will get it definitely. So what is nucleic acid? So first of all, we can write these are basically above are basically scientific terms, so it might be difficult. So this will be the very easy words. These are polymers of. Everyone knows polymers. You have studied polymer in class 10 mathematics. Yes or no?
Yes, sir.
No, wait a minute. Sorry. I was polynomial that was. I was just confused. Polymers, you have studied it in class 11th that it is from hydrocarbon, how we made polythene when n molecules of ethene combine and there will be a condition, then it will be converted into polythene. So basically, these are polymers of nucleotides These are polymers of nucleotides present in the nucleus, present in the nucleus of the cell, present in the nucleus of cell. Also, these are also called, these are also called polynucleotides. Polynucleotides. So basically, what is nucleic acid? So we can easily say several units of nucleotides present in the nucleus of the cell, also called polynucleotides, that is called nucleic acid. As for example, we have deoxyribonucleic acid, deoxyribonucleic acid, nucleic acid, also known as DNA. And the next one is ribonucleic acid, ribonucleic acid, that is known as RNA. Please write this. Fawaz, am I audible to you?
Yes, sir.
Okay, so we just started. I'm gonna slide it up. Then you are going to take a screenshot. So we just started from here. What is denaturation of protein? Whenever you are going to change the physical, there will be physical change or chemical change, like temperature, pressure, or chemical change like pH value. The protein will lose its biological activity, that will be known as denaturation of protein. Okay.
Okay, sir.
And as for example, milk converting into curd is the example of denaturation of protein. And if you are going to boil egg, that is also denaturation of protein. Please take a screenshot.
Done,
Done.
sir. Yes, sir.
Now we started nucleic acid, a new part, new topic. Basically, what is nucleic acid? When many nucleotides combine together with a linkage that is known as phosphodiester linkage, that is called nucleic acid. When two or more nucleic acid nucleotides combine together with the linkage known as phosphodiester, that is known as nucleic acid. Is that clear?
Yes, sir.
Take this screenshot
Yes, I took it.
now. And another word we can write. These are polymer of
Sir, it's
nucleotides
RNA.
present in the. Yes, R.
It's
Oh, sorry, R
RNA.
and D. Yes, it's RNA. Sorry. So these are polymers of nucleotides present in the nucleus of the cell with a linkage that is known as phosphodiester, that is known as nucleotides. So here is the example of nucleotides. Sorry, nucleic acid, that is deoxyribonucleic acid, and ribonucleic acid, DNA and RNA. Done. Please let me know when you are done with this.
Yes, sir. Finish.
Finished. Okay. So now this is basically we are going to study about the composition of nucleic acid. Composition of nucleic acid. So first of all, we can write the composition is pentose sugar. Second, we can write phosphoric acid. Third, we can write nitrogenous base like NH3amine, etc. We will see exactly what it is. Nitrogenous base. Here is another important point. In DNA, deoxyribonucleic acid, sugar present sugar present is beta D 2 deoxy beta D 2 deoxy ribose. And here is the second. In RNA, ribonucleic acid, sugar present is beta D ribose. And the most important point here is base present in nucleic acid are base present in nucleic acid, that will be nitrogenous base, basically nucleic acids are adenine, that is A, guanine, that is denoted by capital G, cis, cytosine, that is C, and then uracil, that is U, and thymine, that is T. So please write it. These nucleic acids are of nucleoside and nucleotide. What is the difference between it? We will see. It's very easy to understand. That means for us, tomorrow we are going to start a new chapter that is known as alcohols, phenols, and ethers.
Okay, sir.
Second chapter of organic chemistry.
Done, sir.
Thank you.
Yes, sir. Finish.
Please just wait a minute. I'm doing something for all of you. Nucleic acid. Okay, wait a minute. Just wait for a few minutes, please. Nafisa and all of you, am I audible?
Yes, sir.
Yes,
So,
sir.
It's been a bit laggy.
okay, wait a minute. Uh, Okay, can you wait for 2 to 3 minutes? I'll be back soon.
It's okay now, sir.
Okay, okay, just wait a minute, please.
Okay.
Am I audible? All of you, Sneha, Nafisa, and
Yes,
Fawaz.
sir.
Okay, sorry, I was. Okay, so basically, you can see in base present in nucleic acid are adenine, guanine, cytosine, uracil, and thymine. That is all denoted by respective symbol. Now, in DNA, which type of base present? So, in DNA, adenine present, guanine present, cytosine present, and thymine present. In RNA, adenine present, guanine present, cytosine present, and uracil present. And here we are gonna talk about nucleoside and nucleotide. Please write this, and then I'm going to write it in another slide.
Sir, can you scroll down?
Scroll down or up. Oh, sorry, it was just changed. I had opened a PDF. Okay, let me know when you are done with this.
Done, sir.
Done. So I'm going to share a PDF. Wait a minute. No, wait a minute. Okay. Now see, nucleoside and nucleotide. Nucleotide. So what it is basically, it's very easy to understand. In nucleoside, only sugar plus base present. Only sugar plus base present. And in nucleotides, sugar plus base plus sugar plus base plus phosphoric acid. Phosphoric acid. So if we want to say, we can say like this is sugar, just suppose, and this is connected with base. So base plus sugar, base plus sugar. When we talk about this nucleotide, there will be sugar like this, and also there will be base along with which connected with phosphoric acid. So this is sugar plus base plus phosphoric. Phosphate, we can say phosphate. Please do write it. Then we are gonna distinguish between this nucleotide, nucleoside and nucleotide.
Done, sir.
Done, everyone?
Yes, sir.
Okay. Now see, we are gonna differentiate between this nucleoside and nucleotide. So see, first point we can write is nucleoside is a compound. Hamza Saikhani, why are you so late? Nucleoside is a compound. Wait a minute. What's going on? Please do wait. Can you see the screen?
Yes, sir.
Okay, so I was writing nucleoside is a compound. Write with me. Nucleoside is a compound formed by the union of a nitrogen base with a pentose sugar. With a pentose sugar. Next, what is nucleotide? Basically, nucleotide we can define in the same way. Nucleotide is a compound formed by the union of nitrogen base, a pentose sugar, and and phosphate. So it is only formed by the combination of nitrogen base and sugar, pentose sugar, and it is nitrogen base pentose sugar along with phosphate. Next point we can write. It is a component of nucleotide. It is a component of nucleotide and here what about nucleotide? Nucleotide is formed through phosphorylation phosphorylation of nucleoside. Please write these 2 points. I'm gonna write the third point. It is slightly basic. In nature. What about this? And this is acidic in nature. A nucleotide is acidic in nature. Then we are gonna differentiate between RNA and DNA.
Done, sir.
Very good.
Yes, sir.
What about you, Hamza Sekhani?
Yes, sir.
Okay. So this is the difference between nucleoside and nucleotide. Now write the topic. That is difference between DNA and RNA. Difference between DNA and RNA. So here I'm gonna write DNA and here I'm gonna write RNA. Please write with me. So the first we can write, it is double stranded nucleic acid. Double stranded nucleic acid. What about RNA? It is single. It is single stranded. Nucleic acid. Second, we can write, it contains deoxy ribose sugar, ribose sugar, and it contains ribose sugar. It contains ribose sugar. Next, we can write, it contains thymine as nitrogenous base. It contains thymine as nitrogenous. Please write capital T in bracket. Nitrogenous base. Beside this thymine, please write capital T. And here it contains uracil instead of thymine. It contains uracil. That means U instead of thymine. Instead of thymine. Next, we can write, it is the genetic and I'm going to slide it up slightly. It is the genetic and hereditary material. Hereditary material of the cell. Everyone aware of genetic hereditary? You have studied this in your
Yes, sir.
biology. It is involved in synthesis of protein. The last point we can write, proteins. It is present in the nucleus of the cell. It is present in the nucleus of the cells. And here we can write, it is present in nucleus and cytoplasm. It is present in both nucleus and cytoplasm. Please write it. After that, we will see types of RNA. That is three types. That are of three types: messenger RNA, ribosomal RNA, transfer RNA. And we will see its functions too. And then we will see a structure of DNA and a structure of RNA. Very easy.
Done, sir.
Very good. Everyone done with this?
Yes, sir. Finish.
Very good. Now, types of RNA. Types of RNA. So it's very easy. The first one is messenger RNA, also known as mRNA, M for messenger. Now, what is its function? So basically, this carries genetic code from deoxyribonucleic acid, DNA, to ribosomes. DNA to ribosomes, where protein is synthesized. Where protein is now the next type is basically ribosomal RNA, and is denoted by ribosomal RNA. Ribosomal RNA. So we will see what's its function. This provides site for synthesis. This provides site for protein synthesis. The third one is transfer RNA. That means tRNA. So what's its work? This transfer amino acid from different parts of cytoplasm to ribosomes during protein synthesis. So basically during protein synthesis, transfer RNA transfer amino acid to different parts.
Done, sir.
Done, everyone?
Yes, sir.
Very good. Now I'm going to show you the structure of DNA and RNA. So basically, we differentiate between these two. And you know that this is basically the structure. You have studied this in biology, mitochondria, and you know the function of this plasma membrane, cytoplasm, microtubule, ribosomes, rough endoplasmic reticulum. But we just don't need to focus in chemistry here. We need to focus on the structure of DNA. And the RNA is not that much important. The structure of DNA, you can see DNA has double helical structure with ACT. ACT, you know, adenine, cytosine, and thymine, and GCC linked together through 2 and 3 hydrogen and respectively through 2 and 3 hydrogen bond. So you can see this is adenine, this is thymine, this is cytosine, this is guanine, and thymine and adenine linked through 2 hydrogen bond, and guanine and cytosine linked through triple hydrogen bond, and this is basically phosphate backbone. So you can write this structure of DNA and take a screenshot and let me know.
Yes, sir. Finish.
Are you done with this, Fawaz, uh,
Sir,
Nafisa?
draw करना है?
You just take the
Sir.
screenshot of this. Okay, yes.
Okay.
Yes, sir. Finish. Take a screenshot of it.
Okay, okay. And please write it in your notes. Okay.
Okay, sir.
And this is not that much important, but you should take a screenshot. The structure of RNA.
Yes, sir.
So
Finish.
here are complete. This chapter is completed. So wait a minute. I'm gonna give you some important questions. So after this chapter, which chapter we're gonna start? Alcohols, phenol, and
And
ethers.
ethers.
Yes. Last test of all of you was quite well. Sneha, were you present in the last class, the last test?
No, sir. I was not present in the last two classes.
Okay. But you should. Wait a minute, I'm gonna. So in the last class, we had done questions on carbohydrates only. It was basically. So I'm gonna take on the question. You just don't need to write the question. You need to write the question number and write the answer. Okay, like A, B, C. Are you
Yes,
getting
sir.
my point?
Yes, sir.
39, 40, 41. Each question carrying half marks. Okay, so there will be. I'll see how much question I'm gonna give you. There's 4 questions first.
Sir, what is this?
This question is for test, Hamza. We have to update every week. Okay, Hamza.
Sir, finish.
Yes, very good. Can I scroll down, everyone?
Yes,
Yes,
sir.
sir.
Okay, two questions. Please write the question number as it is, 4350. What about you, Fawaz?
Yes, sir. Done.
Sir, can you get scroll to previous question? I need to write down because I was not in the meeting.
This is 4 questions from here. Please do it and let me know.
Sir, the previous question. Yes, this one. 1 minute. Okay, done, sir.
Okay, these 2 questions are from here, 43 and 50.
Okay.
Wait a minute, please. Yes, amino acids. Am I audible to all of you?
Yes, sir.
45, 46, 47, and then 52. Very good, Sneha. Just wait for few minutes. Other students are doing.
Yes, sir.
Yes. Okay. Yes. Nafisa, you are also done with this.
Yes, yes, sir.
Fawaz.
Yes, sir.
Okay. Please do this 49 and 54 as well.
Yes, sir.
Done, sir.
Everyone. Hamza, Sneha,
Yes, sir. That's
very good. So how many questions it is now?
12 questions.
12, 13, 14, and this 58. Only these three questions.
Done, sir.
Very good. Send it to the group, your answer. Everyone done with this?
No, sir. Yes, sir. Finish.
Okay, you can send it to the WhatsApp group you are added in.
सर वो ग्रुप में मुझे नहीं ऐड की
You will be added. Please tell whatever, whichever number you have on WhatsApp. Okay.
ओके सर
Everyone done with this?
Yes, sir.
Okay, you may leave the meeting.
Thank you, sir.
Thank you, everyone. Thank you.
Sir, I will send the answers after my father returns because he's not at home. I cannot send you the picture.
Okay, okay.
Okay, sir. Thank you. Assalamu alaikum.
Thank you.